So we spoke a little bit about difficulty of finding, say, ADME data that's been gathered under controlled and similar experimental conditions. As we get into our therapeutic programs, right, we're looking beyond, say, the affinity of our molecules for targets and their functional activity, maybe against those targets, to look at selectivity against related or unrelated targets, ADME properties, physical chemical properties, toxicity. How do people go about choosing how to add predictive models as partners for a larger suite of properties? And how do those decisions and work to build those models, incorporate them, play alongside experimental screening against panels for safety, screening for, say, GPCRs or kinases that often happen? How does one make the decision, right, to expand models when you know you may be introducing some more noise into your predictions around your experimental series?
In our pipeline, target-based, we expand a lot with synthetic data and simulations. So, for example, we have a target that is an enzyme that is part of a big family of enzymes. There are 12 proteins of the same enzyme, and then you have to translate to the animal models, so you have the animal version of it. And so we, for the design of selective, because we are interested in one single enzyme respect to the family, then we do the virtual screen on the entire family.
Okay.
So we try to model the complexity and the selectivity within the very start of the design of the molecule. And now it's possible with the computer power and the simulation. So at the end, you can have 50 targets in the system, even if you are targeting one single experimental, and then we sample the selectivity on steps, and we don't have to do experiments. So we save money and time, and we start from the design. So the molecule, the design do take into account selectivity at the very beginning. So I think that is moving very fast, the idea to complement predicted data along with experimental data that can be combined and then validated, and then you remodel and reuse this data to optimize, as I said, in a cycle. And it's actually what we do. So we embody in the model the selectivity data and eventually the crystal structure when we find molecule. And then we redo all the models, and then we do the docking. And so the next cycle, we take into account new binding mode, strange molecule, side effect to change part of the pocket. So it is integrated. And so we can drive up to 12 very close enzymes moving to selectivity.
Sorry, I was thinking this is the trend actually that is picking up. Of course, it's somehow confined or like played by the biotech companies that do have at least in their specific expertise, physics-based modeling. And in that sense, yes, it's becoming today is much cheaper to, once we have a list of potential candidates, to do our own testing of binding with specific ion channels. You don't have to go for toxicity. You can do it in-house by testing it in silico before even synthesizing and doing it. So there are some room for embedding inside some parts of the risk-taking in which we try to lower it down. Again, it's somehow, I would say, done by companies, large or small ones anyway, that do have physics-based modeling. And it's not only, this is not data, you really play with the players. If you mentioned docking, you can go all the way to affinity and binding, and you can test whether your target binds to hERG channel, is it going to be toxic or not, or to a lot family. The cost is really low. You can do that within a day. In calculation, you have resources. So
this is excellent, and it brings then what the question is, like, how do you decide at some point you want to go and actually verify, and so how many cycles, in silico cycles, would you go through before you say, look, we really want to verify across these 12 enzymes, across the hERG channel, in the laboratory? It's more than we would in the past, right, because you have confidence. but you should go and put experimental stakes in the ground just to verify that and give you confidence in driving forward, it seems to me.
Yeah, and along those lines, we're looking at a multi-parameter optimization problem, right? And if our models, we know they're not perfect, how do we do our weighting on all our parameters when we decide to make molecules? Lots of people use Pareto optimization. There are other ways to do it, but how do we balance the prediction with your experimental resources?
It's a good point. For the same exercise of the enzyme, we decide, in fact, to generate the entire set of the family with the first round. That, of course, cost a lot, but give us the grounds of the model. We also generate one crystal structure for the worst anti-target that we have. So just to make sure, because we don't want to touch something that is critical for toxicity. The rest, we can handle with. So in this case, we generate the crystal structure of the anti-target with the same family, and we model apart.
Nice.
Because we don't want that one, and we can handle the other one.
Yeah.
So you're completely true, and this is the mindset. So you have to go and say, I need data, because I need local models that support the design. So you have to generate this data that I don't need, because the molecule is not at that stage. But it will be faster to that stage if I generate the right model at the very beginning.
Yeah, I agree.
So I think it's getting the balance right with doing the amount of in silico versus the amount of lab testing, because are you ruling out some compounds that might be good compounds that they have bad predicted properties? When you test them, they might not have those properties. So I think it's just getting that, you say, that balance right.
And now the system is evolving very fast. If you see the explosion of building blocks in the last year.
Yeah.
Because now that you can rely more on simulations, then what is missing is the regions. So there is an explosion of building blocks, novel chemistry, novel functional group. So every week it will appear a new functional group in the arena. So because now that you can design with a certain level of confidence, then you need building blocks. Otherwise, you spend too much time in generate the molecule that you designed. If you see, really move to hundreds of thousands up to millions of regions today, that then is the basis of the generative chemistry momentum that is growing fast, that is driven by the fact that the chemical space of region is exploded in the last 5 years, I think.
Yeah, just the interesting thing is then how quickly can we follow up with the chemistry? And could one consideration be like, oh, we can predict this is 6 steps to this molecule, or it's going to be 6 weeks or some time. Say that time is better spent doing another cycle in silico and assuming the risk, right, that of what we see. While we make that, we should be going further. So we're assuming that it'll be correct, so that in fact we're not wasting time waiting for that. And waiting is, it is
It's almost like parallel asynchronous optimization, right?
That's interesting. Okay. And so we're talking about these as processes that are showing themselves to be and can be more productive in time sense and cost sense and a confidence sense. But what we're really talking about is how discovery scientists and medicinal chemists will be working going forward, right? With their agents and their colleagues who are helping them to discover these molecules, design these molecules. What might things look like for them, for us, 2-3 years from now?
Very tough question in a so fast evolving model. But I think that we will see a real the impact of the generative chemistry and the assistant very, very fast. Even if the data that you find in the literature are really noisy, there is possibility to extract information. For example, we have an exercise that we are trying to train a generative model to understand structural activity relationship based on millions of data. So the point is that nobody can have in his mind the relationship on 1,000 targets. But then it's immediate because when you design a molecule, you say, it looks like this and give you the close analogs. So you have immediate adjust because you don't want to touch something. You are moving in a space that is different. If you do offline, then you have to test and think. But when you design the molecules, do appear the information that you are entering in the structural activity of something, or you are close to, or you are moving to an anti-target like ERG immediately, then it changes a lot in life cycle because you don't make the error because you don't have the information. So the difference is that now we ask for something. The future will be we take advice during the design phase. Because if you ask, you need to think there is a problem and search for the problem. The reverse is that I suggest you that maybe something is wrong with this molecule for this reason. And it changes completely because you don't need to think any possible threat. It's the system that is supporting with the right advice. And this is the transformational thing that I think will happen in 5 years from now.
If the question is for a CRO, standard CRO on the bench, I would say this is great news, because what's happening is that a lot more data would be produced upfront by people who have been investing time and going to have a sense of believing that their data is worth a lot. And there is no shortcut. They cannot, nobody today, I mean, except if it's foolish, can consider they're going to be immediately from computer to clinical trials, right? They have to go through CROs. So that means you will have more work to do because hundreds of biotech companies are going to claim because they have this new tech developed, they will have more insurance a little bit closer to real optimized hits, and they will have to do the bench work. They will have to go confront the predictions, because we can't lie to each other. Even if it's physics-based, it's still only models with the energies that are estimated or calculated based on so many assumptions. I mean, the computer model is just a model, and basically they're all wrong.
Sometimes they're useful.
Sometimes they're useful. So we should never forget that. And so what I would guess is there will be a flood of potential candidate where people will come and swear that this is the magic one that will work against this or this target. So it's a bright future for bench classical CROs. Except that we cut maybe from our side, if we are generating potential candidate, the risks by pushing a little bit harder what we can use in silico, AI, you name it, upfront to lower the risks of having to pay big money for.
Sure. It turns out that, the synthesis of a single molecule now becomes, in a fractional sense, a real investment.
I mean, what you said is really that each molecule becomes a multivariate hypothesis, right? That's put into the system and tested, and then adds to the system from a data perspective when it's measured.
The annotation of each and every one of these molecules has gone up by log.
You're screening less compounds, but you're generating more data on the better compounds. Yeah.